Welcome everyone, and thank you for joining today's panel on Looking to the Future, What's Next in Antibody Therapeutics. My name is John Proctor. I'm the Senior Vice President of Antibody Therapeutics here at Berkeley Lights, and I'm joined today by two of our customers, Tracey Mullens of Abveris and Brian Walters of Genovac. And together we're going to discuss the evolving landscape of antibody therapeutics and the technologies that are driving the innovation in this space. And so to get started, I'd like to ask each of you to introduce yourselves and your organizations. And if I could, I'll ask you to go first, Tracey.
Sounds great. Thanks, John. So my name is Tracey Mullens. I'm the CEO at Abveris. Abveris is a Boston-based antibody discovery partner. We specialize in accelerated antibody discovery for both critical reagents and therapeutic antibodies.
Awesome. Brian?
Hello, John. Thank you. I'm Brian Walters. I'm the CEO of Genovac Antibody Discovery. We're a contract manufacturing organization specializing in genetic immunization as an approach to rapid, robust antibody discovery with operations in Fargo, North Dakota and Freiburg, Germany.
Awesome. Well, it's a pleasure to have you both. And I think it's an exciting time for antibody therapeutics today. And the last year has certainly driven a lot of change in this space. And so I'd like to just start by asking, what are the trends today? I think you guys are both well positioned. You have a lot of customers now. What are the trends that you're seeing in the antibody therapeutic space?
Sure. Would you like me to go first?
Yeah,
Brian?
go ahead, Tracey.
Okay, sounds good. So yeah, I mean, obviously, I would agree with you for sure. We're definitely seeing a lot of really exciting new trends within the biopharma discovery space. I think in looking at the space as a whole, it's really kind of hard to pick, what are the primary trends that we're seeing. But if I had to kind of narrow it down or I guess bucket them, I would say that I'd see a lot of trends with respect to target classes, and then also a lot of trends with respect to new technologies. And in fact, I think those evolutions are pretty synergistic in nature. So with respect to targets, I mean, obviously, I think we can all agree that the targets are becoming more challenging, right? We hear people talking about, the high hanging fruit all the time. Basically, what that means is that the readily tractable targets are really no longer really part of the modern therapeutic landscape. So now what we're looking at are, more complex multi-pass transmembrane proteins, GPCRs, ion channels, things like that are obviously really challenging to work with from an antibody discovery perspective. They're hard to generate reliable recombinant proteins. They can be really challenging to generate cell lines. And on top of that, finding functional antibodies for these types of targets can also be even more challenging, which really kind of necessitates function-forward screening workflows. And obviously, we're seeing a lot of increased risk with each of these projects, right? And I think another big trend we're seeing is overall increase in expenditure associated with biologic preclinical discovery. But on the flip side, I also note some really exciting sort of constant innovations with respect to new technologies. And I think that that's a really exciting trend that's going along with the increase in complexity with some of these targets. So new technologies are answering things like, how do you make reliable recombinant forms of the protein, or how do you effectively screen them? And that is probably the most exciting new trend that I've observed and it's funny too, that's actually it can be a challenging trend for a service provider to kind of stick with, right? Because we're always prioritizing our partner programs over other programs. But, it's really important for us, in order to stay at the forefront of antibody discovery, to continue to evaluate these new technologies and kind of prioritize tech development as well. And so one of the new trends, I guess the last one I'll say here, that I've noticed with respect to new technologies is that oftentimes now we have partners coming to us and saying, hey, have you heard of this cool new technology? Can we co-develop this with you guys at Abveris? And that's really exciting. It's an exciting time to be at an antibody discovery company, because you get to do all of that really cool new tech dev along with the partners that are working in the field.
Yeah, that's fantastic. And I definitely want to circle back to to your comment about co-developing new technologies. I think there's a lot going on there. Brian, but let me give you an opportunity. What trends are you seeing through your interactions with customers at Genovac?
I would definitely align with Tracey's response, especially for us as it relates to new technologies. So we have new enabling technologies, powerful technologies. The Beacon itself from your company, John, It's just changing the way that we approach and redefining what's possible, I think, within antibody discovery. For us, the targets haven't really changed that much. Our company was founded on genetic immunization technology. So our clients come in really two different classes. It's a lot of large biopharma, biotech who have their own antibody discovery departments who send us all their GPCR9 channels, and they've been doing that for many years because of that powerful genetic immunization technology, where we can really quickly make DNA for almost all these targets, which is a little different than the challenges associated with making proteins. So the targets haven't changed for us that much, but the tools from which we can now screen and find and discover those antibody producing cells has substantially changed, and speed, I think, is another big fundamental change, that both with what's enabled with the technology that we're using in the tool sets we have, but we're witnessing the largest clinical trial in the history of the planet with COVID vaccinations here. By the end of the year, 1 billion people will have been participating in this with an mRNA vaccine platform. So I think speed has got to be redefined here in terms of what's possible. I don't believe there was a mRNA vaccine that had gone to a phase 3 clinical trial after many years of development. So I think that's about ready to change rather significantly. And then the modalities, I think that the different approaches for not only antibody therapeutics, but antibody-based therapeutics and the multispecifics, the encoding into mRNA vaccines, DNA vaccines. I think the modalities and what our clients are doing with the antibodies that we discover for them is changing quite a bit.
Yeah, for sure. And your comment about the largest clinical trial, and it's not only challenging, but it's really exciting. And I think it's, as you said, Tracey, it's exciting to be a part of this rapid change that is taking place. And I know for both of your companies, there's been a lot of change. I mean, COVID aside, I think both of you have gone through a significant amount of change over the last year. Can you talk a little bit about specifically what you've had to change internally to keep up with the speed, as you say, Brian, and all the new challenges that COVID has brought? And Brian, I'll let you start with this one.
Oh, it's on a lot of fronts, for us. I think the adoption of new technologies in itself has been, big focus of our company. New capabilities, new services. We work again from that genetic immunization platform, so we're constantly looking at new technologies that are sort of protein free, if you will, for time saving, expense saving options. So that's been definitely significant. I think immediately last year we saw the need to take our safety stocks and triple them, just because of supply chain constraints. I mean, even some simple things, almost anything plastic and pipette tips are sort of the industry's toilet paper, it feels like right now, and there's all kinds of back orders, and so I think managing that while growing at the same time has been a challenge, and it's been I think a lot of fun watching the field respond and come together and work together in different ways. We're seeing large pharmaceutical companies who are definitely competitors cooperating on manufacturing right now. So I think it's opened our mind to a lot of new, innovative, creative approaches from which to pursue the industry's opportunities and challenges.
Yeah, great. Tracey, how about for you guys? What have you guys had to change to keep up in this fast-paced environment now?
Yeah, we've definitely done a lot of made a lot of changes, honestly. But I guess some of the main ones to point out, obviously we're, recently announced that we actually brought on a second beacon to really kind of help with the demand. So one of the things that COVID kind of introduced to our organization is a number of new partners. They had to shut down their own facilities. They needed to outsource. They wanted to keep their workflows going. That was a lot of increased demand. We had we brought on a second beacon. Not only did that enable us to help with the demand, but it also enabled more complex workflows. So we're now able to continue to optimize things like functional screening, making sure that we're screening for the right specificity and functionality up front to help accelerate those antibody discovery campaigns. We've been integrating, kind of back to the new technologies piece there, we've been integrating some new platforms into our single cell workflow, both upstream and downstream, and using some of these platforms in pretty novel ways to kind of help out with that. And then just to make sure that we're fully capturing the full genetic diversity of any mouse model or any other species that we're working with, we started to add things like repertoire sequencing analysis into the workflow, which has really kind of enabled us to, again, capture that full diversity of those species. I guess speaking of, we're actually currently now exploring additional alternative species to also further help with diversity and different antibody formats and things like that. It's too soon to announce the details, but going to be providing some exciting new offerings soon. And then, of course, just expanding in general, expanding our Beacon team, probably should plug that we're hiring, and introducing some downstream engineering capabilities and things like that to kind of help with all of these complexities associated with the new target classes.
Yeah, very cool. I appreciate you both mentioning Beacon as part of what has at least been a part of the changes that have occurred for both of you. Of course, I'm biased, but we believe Beacon really is a game changer in terms of capacity, speed, functional analysis, and those things. So I just wanted to ask for you guys, how has Beacon been able to keep up with the demands that you've had? I think you've both added capacity in this regard over the last year. How has Beacon really enabled you to take on more? Brian, maybe you can kick this one off.
Yeah, no, like as Abveris, we have a second Beacon, and so I think that's definitely not only added capacity, new screening capabilities, allows us to do more sequential assays, and while we continue to work on cell life viability to allow for more of that to occur. So we're screening 80,000 cells in a day, in some cases on a single project. That gives us much broader coverage. The deep sequencing and the repertoire work is critical to the field, and so sequence enrichment, definitely a workflow that we're integrating now as well. New species, we introduced rat and transgenic rat last year, so we're very excited to that. We're seeing great results there. That diversity component that Tracey mentioned is definitely a critical enabling piece for us. So, but we're circling back to some projects that we've always worked on GPCR and ion channels because of our genetic immunization technology. So I think what we're finding is combining that front end technology with the Beacon, complete focus of ours the last 12 months. That really, in terms of moving up the tree of targets that we're now able to have success on. Not only are we seeing significant increases in number of hits, we're seeing success on targets that just failed previously, just because we couldn't find the cells. So we're finding them now, so that's exciting. It's going to unleash a whole new era of therapeutics, and no doubt as a result. So it's definitely had a substantial impact to us, more importantly to our clients, in terms of boosting success rates, and then doing cutting timelines too. I mean, we're saving months of screening and development time with the capabilities of the Beacon.
Awesome. Tracey, I know you've talked about this in some of your other webinars, but maybe you could just expand on how Beacon has impacted you guys.
Yeah, definitely. I mean, I think I definitely echo a lot of what has already been mentioned by Brian, but I think one of the biggest things that we really appreciate about the Beacon, and I'm sure Genovac in general will probably agree with this, is really the ultimate flexibility in assay design. And so one of the things that we really appreciate is the ability to not just interrogate a large panel of antibodies for one or maybe two specificities, but really kind of deeply dive into that screening resolution, look at things like multi-species specificity, cross-reactivity to off-target family members, but then incorporating things like functional screening into the workflow, right? Again, going back to that point of incorporating a function-forward screening paradigm is really important for us to be able to pull forward the most relevant antibodies. And that benefits platforms and campaigns across the board, because again, now you're not wasting valuable time and money and resources recombinantly expressing a bunch of irrelevant antibodies. At the end of the day, you're really only moving forward with the best of the best candidates that are out there. And I think that has really revolutionized our workflow. It's really kind of removed the whole burden from the downstream process. And really, it's not even at the expense of additional burden upstream, right? Because it's just a single day of screening. So that's pretty incredible, and that's kind of revolutionized our entire single cell discovery workflow.
Yeah, that's amazing. I love hearing the stories that you both have, and for those of you that haven't seen them, both Abveris and Genovac have given excellent webinars on the success they've had in campaigns for their customers. So if you haven't seen them, please circle back to their websites and check those out. So as we kind of wrap up the panel here, I wanted to look to the future a little bit and ask a couple of things. So one, you guys have mentioned some trends in terms of targets, technology evolution. Where is that driving you in terms of services that you're going to be offering to your customers in the future? And Tracey, if you'd like, I'd like to ask you to start with this one.
Yeah, I mean, moving into the future, as I mentioned, we're definitely going to continue to explore alternative species, alternative workflows. I think one of the things that we really appreciate about the Beacon and with respect to the assay flexibility is, again, that ability to do some functional screening on chips. So we really have a lot of optimization underway for bringing some of those functional screens online. And we've kind of learned, as we keep getting new challenging targets, that there's really no one size fits all when it comes to functional assay development. So we often do, very separate workflows where we'll just work with a partner and optimize a functional screen and then move forward from there. And that's been really fun for our Beacon team and all the scientists at Abveris. We just love coming up with really cool new assay designs and adapting things that you wouldn't think would work in a fluorescence-based readout, figuring out a way to make that work. It's been really fun. And then just continuing to make sure that we stay at the forefront of discovery through, again, that continued evaluation of new technologies. I think one thing that I really love about our organization is that pretty much everybody here has this shared mission to really make sure that we maintain industry thought leader status and that we're not ever really wasting time with dated technologies and trying to get them up and running, but instead really kind of seeing what else is out there, continuing to evolve with the changes in the industry. And so that's what we're definitely going to continue to do, right? We're definitely going to keep going. We're going to keep evaluating new technologies. We're going to keep bolting on new things to our single cell workflow and then vertically integrating additional offerings as we have the capacity and bandwidth.
Awesome. Brian?
I think much along the lines of Tracey, more investment, more innovation. I think drawing 22 years of experience working on challenging targets and continue to adopt new platforms and combining that experience, our immunization technologies, the screening technologies, definitely a focus on cell-based assays and functional assays as Abveris as well. The field needs us to do that to speed up the development process. In general, anything that helps us improve success rates and do it faster and speed up the development timeline is a focus of our organization. In Fargo here, where I'm sitting, we're adding production and characterization capabilities to complement what we have in discovery capabilities in Germany. Eventually, geographic diversification is part of our short-term mission too. Within 12 months, we'll have discovery and production in both locations for redundancy, more capacity, risk mitigation in some cases. So we're excited about that and the growth that's going to come from that. More species definitely on the horizon for us. We're looking at rabbit now. We're looking at alpaca. Again, I think the field needs us to do that in the formats that from which they would like to pursue their drug candidates. Despite the fact that we have pretty innovative and long track record using genetic immunization, half of the battle is getting those antibody producing cells to put on your instruments. So we're going to continue to innovate and focus in that area, looking at new technologies and mRNA, next generation protein classes and things like that, that are platforms, I should say, that we're constantly continuously looking to improve on.
Yeah, that's amazing. I think, the expansion of species and as, downstream, production, as you said, Brian, and, maybe one of these days we'll have a panel and talk about cell line development, which is a little further downstream, but I think it sounds like you both are perhaps migrating that way, which is great to hear. Is there anything else you'd like the audience to know before we close?
I mean, definitely, if you are interested in doing some tech development, reach out. We really love those projects. Those are our favorite for sure. We love really collaborative partnerships. So that's what I would say.
Yeah, I would say it's, as mentioned earlier by Tracey, it's an exciting time to be in the field. There's new technologies that, in combination of existing technologies, new technologies that are allowing us to do new things. Speed definitely, I think, is about to be redefined globally in terms of the time it takes to take any drug to the market, the modalities and approaches, and to see something like mRNA vaccine advance as much as it did over the past year and a half, and knowing people are working on taking antibody sequences and coding them into mRNA vaccine platforms and the various modalities that will come from that is an exciting time. So we're exploring all these things, and we'll continue to add value to our clients, and like Tracey, welcome any discussions in terms of collaborations and how we can help serve the field better.
Awesome. Well, thank you both so much. It's been a pleasure to be on the panel with you, and as always, it's a pleasure to work with you both. So I think at this point, I'd like to open the door for some live questions from our audience. Thank you both for joining me for this panel. At this point, let's take some questions from the audience. The first question we have here is, what do you think the next challenge will be? Tracey, maybe you'd like to kick that one off.
Sure, definitely. So, one thing that I'm really excited about for the future of antibody discovery is really the incorporation of in silico based workflows into antibody discovery workflows in general. And I think that while that is really exciting, it probably poses a fairly significant challenge with respect to kind of how the industry answers that particular addition to the workflow. So, and what I mean by that is, the big data analysis portion of the data mining for in silico workflows, and then actually integrating that machine learning based AI into campaigns, I think will be probably the next biggest challenge that we'll face. Currently, there really doesn't seem to be a centralized data management system. There's really no census for the ideal biologics-based analysis software that's out there right now. So I think as we as an industry continue to move down the path of in silico-based assessments, being able to actually capitalize on what we're learning from that will probably be a challenge.
Brian, you have anything to add to that?
No, I agree. I think the area of computational biology, electronic biology, synthetic biology, I think the field is changing very rapidly, and I agree with Tracey's comments that that's a big area of change and challenge and opportunity.
Great. Okay, the next question that's come in is, how would you compare the Beacon system to other single-cell technologies or antibody discovery platforms? Brian, can you start?
Yeah, I think for us, the biggest, I think, positive of the Beacon over other B-cell platforms is the ability to do sequential assays. So I think that the sheer robustness, but the speed from which we perform screening and then can flush channels, load new cell lines, and check for beyond binding function cross reactivity, I think is a great feature of the system.
I know you've shared at least a couple of examples where you tried, campaigns both in hybridoma and in Beacon, and in some of those cases you found nothing by hybridoma, but you did find candidates by Beacon. What do you attribute that to?
I think the inefficiency of hybridoma and the ability to screen all the individual cells, or a significant amount of the individual cells, is I think a big game changer. And I think diversity, it seems to be a huge difference. In addition to the yield, we're seeing much greater diversity with the Beacon system.
Tracey, do you want to tackle that question? So how would you compare the Beacon system to other single cell or antibody discovery technologies?
Sure, certainly. So actually recently we had a webinar in which we actually did exactly this. So after kind of reviewing a lot of the different platforms out there and starting to think about the pros and the cons associated with alternative single cell workflows, I think we have kind of bucketed the Beacon system into its separate sort of generation of single cell discovery. And so we refer to the Beacon platform as generation 3, and generation 2 and 1 being nanowell technologies and then encapsulation technologies respectively. And I think that all of these platforms definitely have their own sort of specific advantages, but I think at the end of the day, the biggest limitation in both of the technologies is really the number of screens that you can perform, the fact that you can only typically perform a single no-wash assay, and then the throughput itself. It can be limited by Poisson distribution in the case of the nanowell technologies. And also that makes it really challenging for cell surface screens and also functional screens. And these are all things that the Beacon is able to do in a very unique way that kind of sets it apart from alternative technologies out there. I agree with Brian that the sequential assay benefit is tremendous. We're able to look for specificity, off-target specificity, and then flush things out and look for function. And that's what we're really excited about, and being able to screen things like adherent cells while not disturbing the plasma cells in their discrete microenvironments. I think that's just incredible advantage over alternative technologies, and especially as we look to the next generation of targets where these types of cells are a little bit more challenging to work with in general, that's where the system will probably have a huge advantage over alternative technologies.
That's great. There are more questions coming in kind of around this area. So one of them is, has using the Berkeley Lights system shifted the antibody screening bottleneck, and if so, where is the biggest bottleneck now when using the system?
So
Tracey, Brian, either one of you jump in.
Sure, yeah. So I think that at least what we've been seeing with respect to the bottleneck is kind of in line with what I had identified as, in my opinion, the next challenge within antibody discovery is just the big data analysis, the big data mining. You generate so much data from these screens. You're interrogating tens of thousands, even hundreds of thousands of single plasma cells for a number of different specificities, a number of different assays, and then you need to really quickly and efficiently interrogate all of this data and be able to pull forward the best candidates possible from the thousands and oftentimes thousands of hits that you see. And I think that's probably where the new bottleneck is. But I'd love to hear Brian's thoughts on that as well.
Tracey, I agree with you. We're generating more and more data all the time, and I think that management is a bottleneck. And then for us, I think as well, the downstream production towards cell line development, I think maybe not always a bottleneck, but I think an opportunity for us to speed timelines for our clients.
Awesome. Oh, I think we'll hold it there. There are more questions, so I may forward those to you guys. And please, for those of you that participated, please feel free to reach out to Tracey and Brian directly at their organizations and to Berkeley Lights as well, and we'll try to address any additional questions that we can. But thank you all for joining. Thank you, Tracey and Brian, for being part of this panel, and I'd like to wish everybody a great rest of their conference. Thanks so much.